Welcome back to Chapter 14. In this video, we'll focus on Section 14.3, Mechanisms of Antibacterial Drugs. This section looks at how different antibiotics actually work inside bacterial cells, so which structures they target and why they don't usually harm human cells. By the end of this section, you should be able to describe the mechanisms of action for drugs that inhibit the cell wall, biosynthesis, protein synthesis, membrane function, nucleic acid synthesis, and metabolic pathways in bacteria. And we'll be able to explain how these mechanisms demonstrate something called selective toxicity, meaning the drugs kill the microbes without harming the host. Of the antimicrobial drugs we have today, most of them are antibacterial, and that's because prokaryotic cells have unique targets. For example, bacteria have peptidoglycan and 70S ribosomes. We have fewer drugs to target eukaryotic microbes like fungi or protozoa or parasitic worm infections because we are also eukaryotes. So a lot of those targets we also have in our own cells, and that can increase side effects and toxicity. We also have fewer drugs to target viruses. So we're going to go through each of these major mechanisms in this chapter and highlight key drug classes you'll likely encounter in clinical settings. This figure shows how antibiotics are classified based on their site of action. So each of the colored blocks represents a different target inside of the bacterial cell, from the cell wall all the way to DNA replication machinery. So there are 5 major targets described in this section. We have cell wall synthesis, we can target protein synthesis, we can target membrane integrity, nucleic acid synthesis, and metabolic pathways. All of these are essential processes for bacteria to survive. So by disrupting any of these, we can either stop growth, remember that would be bacteriostatic, or it can kill the bacteria, and that would be bactericidal. And our book provides a nice little table of all of the different drug classes we'll be going over in this section, as well as their mode of action, what they target in the bacterial cell. And these look like the cell wall. Here we're targeting protein synthesis, the membrane, nucleic acid synthesis, and the last 2 are metabolism. These are drugs that block metabolic pathways. We're going to start with the group of drugs that inhibit cell wall formation. And drugs in this group are all bactericidal. They weaken the bacterial cell wall until it ruptures. But these drugs only affect active, new and growing and dividing bacteria, because that is when peptidoglycan, remember the main component of bacterial cell walls, is being synthesized. These drugs would not affect existing mature cells because those cell walls have already been built, but the drugs will prevent these cells from dividing or repairing their walls if there is damage. Luckily, human cells, we don't have peptidoglycan, we don't have cell walls. So these drugs show excellent selective toxicity where they affect the bacterium involved or the bacteria involved, but they don't affect our cells. Out of all of the drugs that target the cell wall, the most famous group is probably this group, the beta-lactam antibiotics. But before I jump into how these drugs work, let's do a quick refresh of the cell wall, because it's been a while. So remember, bacterial cell walls are made of peptidoglycan, and the peptido tells me there are peptides, like these amino acid chains involved. The glycan reminds me that there are sugars involved. So if I look at the cell wall of a bacterium, they zoom in here. These hexagon-shaped structures are the glycan part, the sugar part of the cell wall. And a long time ago, we learned that they're made of alternating N-acetylglucosamine and N-acetylmuramic acid subunits, alternating and repeating over and over again. So we have NAG, NAM, NAG, NAM, and so forth. They remind me of kind of like a brick wall, where the bricks are the NAG and NAM. But peptidoglycan also has that peptide part, so that those little peptide chains are just little short strands of amino acids, and here they have them as these vertical kind of purplish lines that hold the sugar chains together. And going back to that brick wall, the peptide part, those peptide chains remind me of the cement, kind of that glue in between the bricks that holds everything together. So how this drug or this class of drugs, beta-lactams, work is they prevent the formation of the peptide chain portion of the wall. So it does that because beta-lactams have this specific chemical structure. The beta-lactam ring kind of looks like a square ring. I'll show you in a bit. And this blocks the enzyme transpeptidase, whose job is to lay down those peptide chains. So sometimes this is also known as penicillin binding protein, transpeptidase is. And if we look here and we pretend that these green dots are beta-lactam antibiotics, they are preventing the formation of the peptido part of the peptidoglycan by blocking this enzyme and preventing the formation of those peptide chains. Now imagine it's the same thing as having this brick wall, but without any of the cement holding the bricks in place. So if I just laid a bunch of bricks on top of each other, it's very easy to knock down, right? I could just poke it with my finger, the bricks would fall. The same thing would be true for our cell walls. Without those peptide linkages, the cell wall is very unstable. Water could enter the cell and cause lysis or bursting of the cell. So this is the drug penicillin, and here's an example of what that beta-lactam ring looks like, this square kind of shaped ring. Unfortunately, there are some bacteria that have resistance to penicillin, and microbes that are resistant because they have enzymes that can destroy our drug, destroy the ring here. And those enzymes are sometimes called or referred to as beta-lactamase. So beta-lactamase is an enzyme that would cleave or destroy my beta-lactam ring, and I can see that here. Now this penicillin has been inactivated. Sometimes these enzymes are also called penicillinases. So what do you think? A bacterium that is beta-lactamase positive or penicillinase positive, is it going to be sensitive to or resistant to penicillin? So it's going to be resistant to penicillin, right? So if you're a bacterium and you have this enzyme, you can break down penicillin and it's no longer active. It can't do anything to you. So now you can survive. You are resistant to that antibiotic. So the classes of beta-lactams include our penicillins. We have natural and semi-synthetic forms, such as amoxicillin and methicillin. Methicillin is a semi-synthetic penicillin that was developed to address the spread of enzymes that resistant bacteria had, like those beta-lactamases or penicillinases that were inactivating the other penicillin types. Cephalosporin, there are several different generations that have been created, and with each subsequent generation, there's an increase in the spectrum coverage so that these drugs can even kill gram-negative bacteria in addition to gram-positive. And in their fifth generation, these drugs can also kill MRSA. So these are methicillin-resistant Staphylococcus aureus. Our monobactams, like aztreonam, these are narrow-spectrum beta-lactam antibiotics. They're effective against gram-negative only. And then we have our carbapenems. These are the broadest in spectrum, and they're effective against many bacteria that have beta-lactamases. This figure shows our beta-lactam antibiotics, and if I compare our natural versus semi-synthetic penicillins, we can see that by adding an amino group to penicillin G, for instance, here's my amino group, and my core structure that I did see in penicillin G. By adding these amino groups, we can increase the spectrum of these drugs against more gram-negative pathogens in addition to those gram-positive. And then by adding that additional hydroxyl group here for amoxicillin, that increased the stability of the drug, especially in acid or acidic conditions, which allowed for improved oral absorption. For methicillin, changing the R group to a bulkier side group over here, side chain, provided protection of the beta-lactam ring from enzymes and penicillinases, giving us the first penicillinase-resistant penicillin. And then if I look up at the top, I can see that cephalosporins, for instance, they're fused to a six-membered ring or six-sided ring, as opposed to penicillin that has a five-sided ring fused to the beta-lactam ring. By having this six-sided ring, we have an increased resistance to beta-lactamase. And it also has 2 R groups rather than just one, and this gives greater diversity for chemical alterations because you could alter this R group or this Al group. So overall, we can see that different R groups can modify the stability of the drug, the activity of the drug, and even the resistance profile of the drug. Our next set of drugs that affect the cell wall include glycopeptides and bacitracin. Vancomycin is a member of a class of compounds called those glycopeptides. And vancomycin is a large glycopeptide that binds directly to the peptidoglycan peptide chain. And this blocks both transglycosylation and transpeptidation. So transglycosylation, this was the process of building those NAM, NAG repeating units of the sugar portion of the cell wall, right? It kind of looked like that earlier in my picture. Transpeptidation, remember those are the peptide links that connect the chains, so from one to the next. And vancomycin is effective only against gram-positive bacteria, including methicillin-resistant Staphylococcus aureus, although these days there are even a class of bacteria or groups of bacteria known as VRSA, vancomycin-resistant Staphylococcus aureus. Bacitracin blocks the transport of peptidoglycan subunits across the cell membrane, and it's used topically on the skin, such as in Neosporin, due to kidney toxicity if it's taken internally. These days, however, many dermatologists, the trend seems to be that dermatologists are not recommending or do not recommend Neosporin due to allergic skin reactions, limited benefits, and also due to contribution of Neosporin to antibiotic resistance, especially in S. aureus and Staphylococcus aureus and Pseudomonas aeruginosa. So this is what I was trying to sketch again earlier. Vancomycin is a big glycopeptide, so it blocks the formation of the sugar chains, the NAG, NAM repeating subunits, but it also is so big that it blocks transpeptidation as well, so we cannot form those linkages, and you can again have decrease the integrity of the cell wall. Whereas bacitracin blocks the transport of peptidoglycan subunits, so cell wall subunits, across the membrane. So if I look at this bacterial cell, here's the membrane in green, and the subunits would be built inside the cell, right? Let me change colors. I need something to stand out. No, that doesn't work either. Let's try yellow. So subunits would be inside the cell, and then you need to transport them out to build the cell wall, because the cell wall is an outer layer that's outside of the cell membrane. So if bacitracin is blocking that transport, then you cannot build a new cell wall. Our book then provides us with a giant table of all of the drugs that inhibit bacterial cell wall synthesis. So we saw that some of the drugs, like penicillins, they inhibit transpeptidase activity because the beta-lactam ring in these penicillins looks very similar to what transpeptidase usually binds to build a peptidoglycan. We also have the cephalosporins. Remember, these are also inhibiting transpeptidase, and these had several classes or several generations, where with increasing generations, we have greater spectrum. So here is my third, fourth, and fifth generations, and you can see as you go down, there is a bigger or broader spectrum. We also have our monobactams and our carbapenems, again, with the last one being the broadest spectrum, and these are effective against even some bacteria that are resistant to most beta-lactams because they have that beta-lactamase enzyme. We also have our glycopeptides like vancomycin that we just saw that can impact or kill gram-positive bacteria, including some MRSA. And then finally bacitracin, which prevents the transport of peptidoglycan subunits across the cell membrane to that outer wall region. So this checkpoint asks us to describe the mode of action of beta-lactams. Beta-lactam antibiotics included penicillins, the cephalosporins, carbapenems, monobactams. So these all have a beta-lactam ring, that square-shaped ring that binds to and inhibits penicillin binding proteins, those PBPs or transpeptidase enzyme. These are enzymes that build crosslinks in the peptidoglycan cell wall. So these drugs weaken the cell wall and they can lead to cell lysis due to osmotic pressure, right? Water rushing into the cell, causing bursting of the cell. So these drugs are bactericidal and they're selectively toxic because human cells don't have peptidoglycan, so they can kill the bacteria without harming us. Now we'll switch gears and look at drugs that inhibit protein synthesis. In this case, ribosomes will be our target because remember, ribosomes are the organelles that build proteins. They are involved in translating messenger RNA and producing our amino acid chain. So bacteria have 70S ribosomes that are different than most of the human ribosomes, 80S. Although there is an exception, remember in our mitochondria, in human mitochondria, we do have some 70S ribosomes. In bacteria, these 70S ribosomes are made of two subunits, the smaller 30S subunits and the larger 50S subunits. This structural difference between most human and the bacterial ribosomes allow drugs to selectively inhibit bacterial translation. But there are some side effects because of our mitochondria, since we do have some 70S ribosomes in our cells. So again, we're looking at inhibiting protein synthesis, and this is kind of a refresher of translation. Remember, in translation, what we're doing is reading messenger RNA that has A, U, G, or C nucleotides, and we have our ribosome with a small subunit binding here, our large subunit, and then our transfer RNAs bringing amino acids to that growing chain. All right, so let's look at the drugs that inhibit protein production by looking at our 30S subunit inhibitors first. These are drugs that inhibit that small subunit of the ribosome. For our aminoglycosides, these include streptomycin, gentamicin, or neomycin. These drugs cause mistranslation of messenger RNA, and this will lead to faulty membrane proteins and cell death. So these are bactericidal, but they can cause, unfortunately, kidney, ear, and nerve toxicity and damage. Our next group are tetracyclines. These were originally naturally occurring, produced by various strains of Streptomyces bacteria, and these were first discovered around the 1940s. Later on, we had several semi-synthetic tetracyclines produced, including doxycycline and tigecycline. These block tRNA, they block transfer RNA from binding, so they prevent peptide chain elongation, and these are bacteriostatic, broad spectrum, but there are some side effects, including light sensitivity, liver toxicity, and tooth discoloration that's permanent. So here we see those aminoglycosides and tetracycline affecting the 30S small ribosomal subunit. Aminoglycosides were the ones that caused mistranslation of messenger RNA, leading to faulty membrane proteins and cell death. The tetracyclines were the ones that prevented or blocked the binding of transfer RNA, and thereby you could not grow your amino acid chain, inhibiting protein synthesis. But one potential side effect of tetracycline that I mentioned is tooth discoloration, and this would be, it's usually permanent, but usually not going to happen in adults. The highest risk is going to occur during tooth development in children. So a lot of physicians will avoid prescribing tetracyclines if or in groups like pregnant women or breastfeeding mothers or children, such as children under 8 years old of age. All right, now let's look at our large ribosomal subunit inhibitors, the 50S subunit inhibitors. We have our macrolides, such as erythromycin and azithromycin. These block peptide bond formation. Remember, peptide bonds are the bonds that connect amino acids. These are broad spectrum bacteriostatic drugs that are luckily well tolerated by the body, and azithromycin in particular is one that has a pretty long half-life. Remember, half-life is how long it takes to clear 50% of the drug from the blood or the plasma, which often means a shorter therapy time. Our lincosamides, such as clindamycin, have a similar mechanism by blocking peptide bonds. They're effective against gram-positive bacteria like streptococci and staphylococci, as well as anaerobics, anaerobic bacteria, but sometimes there's a risk for overgrowth of C. diff, Clostridioides difficile, because those bacteria make endospores. Chloramphenicol is a broad-spectrum drug that also inhibits peptide bond formation, but there is usually limited use only for severe infections because it can also cause aplastic anemia, where we have a deficiency of all types of blood cells. Finally, we have the oxazolidinones, like linezolid, and these block formation of the initiation complex. Remember, that is when we first get everything together, the messenger RNA, the small subunit, that tRNA, and the large subunit. And these are a newer drug class that can actually work against MRSA, methicillin-resistant Staphylococcus aureus, as well as VRE, vancomycin-resistant Enterococcus bacteria. So if we go back to that previous figure we looked at together, the 30S drugs block or affect decoding of the messenger RNA, and they affect tRNA binding as well. Whereas the 50S drugs, they affect peptide bond formation and formation of the initiation complex, where you get everything, all of these components together. And both of these prevent, so both the 30S and the 50S drugs prevent essential protein synthesis and thus bacterial growth. Our book also summarizes this with our table 14.3, showing that drugs that target the 30S subunit affect decoding of the codons on the messenger RNA, and they also affect transfer RNA binding. Whereas those 50S subunit drugs, those inhibitors, affect peptide bond formation between amino acids and also prevent the formation of the initiation complex. So this checkpoint asks us to compare and contrast the different types of protein synthesis inhibitors. And we kind of just saw that in the tables on the previous slide, but I also created a kind of simplified version of that. And here's the simplified version of the table where we can see the 30S drugs that cause misreading of messenger RNA or blocks transfer RNA binding. And then here are the 50S drugs that block peptide bond formation or the initiation complex formation. Now we'll switch gears and look at drugs that inhibit membrane function in bacteria. So drugs that disrupt bacterial membranes, they tend to have poor or low selective toxicity because human membranes are similar in structure. So these drugs are effective, but they're used cautiously or topically because they can also affect our cells. Our membrane targeting drugs include polymyxins, such as polymyxin B, E, or colistin. These bind to lipopolysaccharide, remember that LPS layer, or phospholipids in gram-negative outer membranes. This disrupts the membrane integrity, causing the cell contents to leak out of the cell and eventually cell death. So these drugs are usually used topically or as last resort IV, intravenously, for resistant infections due to kidney and nerve toxicity. For daptomycin, this inserts into gram-positive membranes, including MRSA, methicillin Staphylococcus aureus. These drugs cause membrane depolarization, which messes up the ionic balance inside versus outside of the cell and leads to cell death. And these are usually treated for, in terms of IV use, for skin and blood infections. Table 14.4 summarizes this with our polymyxins interacting with that lipopolysaccharide layer or outer membrane of gram-negative bacteria, and our daptomycin, which is a type of lipopeptide which inserts into the cell membrane of gram-positive bacteria. How do polymyxins inhibit membrane function? So polymyxins, such as polymyxin B, E, or colistin, they kind of act like detergents. They bind to lipopolysaccharides, remember that LPS, and phospholipids in the outer membrane of gram-negative bacteria. This disrupts the integrity of the membrane, leads to increased permeability and leakage of the cell contents and cell death. So these have, unfortunately, low selective toxicity because they can also damage our human membranes. So they're mainly used topically or as a last resort case or drug for multi-drug resistant infections. Our next group of drugs target nucleic acid synthesis. So they're going to target bacterial RNA or DNA enzymes, exploiting the structural differences between bacterial and human polymerases. For our RNA and DNA inhibitors, we have our rifamycins, such as rifampin. These inhibit bacterial RNA polymerase and blocks transcription. This is used in tuberculosis therapy, always in combination with other drugs such as isoniazid or streptomycin, and there are side effects though. So side effects include liver or hepatotoxicity. Rifampin can actually induce liver enzymes to increase the breakdown of other drugs that are being administered at the same time. So this is an antagonistic effect, and this can lead to the liver toxicity, and it negatively influences the bioavailability of the companion drugs. We also have our fluoroquinolones, such as ciprofloxacin or levofloxacin. These inhibit DNA gyrase and topoisomerase 4, and this thus blocks DNA replication in bacteria. So these are broad-spectrum drugs that are often used for urinary tract infections or to treat respiratory or skin infections. Potential side effects include tendon rupture, especially in older adults or those on corticosteroids. So the tendon ruptures may be likely due to degradation of collagen and the inhibition of tendon cells proliferation. Can also have neuro or cardiotoxicity. Finally, we have metronidazole. These are activated in anaerobic cells, and this damages the DNA of the cells, but also can be used against protozoa. Table 14.5 summarizes those two groups. We have rifamycin that inhibits bacterial RNA polymerase, so now we cannot read the DNA to produce messenger RNA, thus blocking transcription, and our fluoroquinolones that block DNA gyrase and topoisomerase, so you can't open up your DNA, you can't separate the two strands, and this blocks DNA replication. This checkpoint asks, why do inhibitors of bacterial nucleic acid synthesis not target host cells? Because we also have DNA and RNA. It's because these drugs target bacterial-specific enzymes that are not present or very structurally different in human cells. Like the fluoroquinolones, they inhibit bacterial DNA gyrase and bacterial topoisomerase. We don't have those particular enzymes in us. And rifamycins, they inhibit bacterial RNA polymerase, which is different in structure from the eukaryotic version. So based on these differences in the enzyme structure, these drugs show selective toxicity, killing bacteria without interfering with human DNA or human RNA synthesis. That takes us to the end of our second video. In our next video, we'll continue our discussion with our inhibitors of metabolic pathways.